Thanks, Aman. It's a pleasure to be here. It was a great introduction. Thanks a lot. I want to thank Aman and Bruce, who's not here yet, for hosting this. So this is a really cool opportunity for me, especially at this stage of my career, to kind of show you guys what I've been working on for the last 2 or 3 years. And so primarily what we're looking at are methods of looking at brain function in mice. So normal mice and then mice under some type of disease model. So Alzheimer's and stroke. And then the last half of the talk is going to be about stroke recovery and then manipulating stroke recovery. So at the risk of telling you things you already know, I'm going to do it anyway. So the brain's functionally organized into distinct regions. So as you listen to me speak, your auditory cortex is active. As you look at my slides and look at me, your visual cortex is active. As I speak to you, I'm incapable of not talking without moving my arms. So my motor cortex is moving and active. And so there's this very distinct functional parcels in the brain. And kind of the way that this was observed and recorded is traditionally with fMRI. Some PET, but mostly fMRI. It's kind of the standard clinical tool now. And so traditionally, any kind of functional neuroimaging study involves putting somebody in this bore and then having them tap their fingers or flashing lights in their eyes, some type of task-based stimulus design. And then while they're undergoing that task, changes in neuronal activity are measured. And so typically, let's say you're flashing a checkerboard in your eye, you'll see changes in the bulb response associated with that task. So if you were to be in the MRI bore and then be asked to open and close your eyes at fixed intervals, you'd see changes in the bulb signal that kind of oscillate with the task paradigm. And so if you subtract the open sessions from the closed and then look at where that maps to, you'd see an activation in visual cortex. So these traditional kind of functional neuroimaging task-based studies were used to relate brain topography to function. But if you look closely at this purple line, it looks noisy. And so almost 20 years ago, that quote-unquote noise over a very specific frequency band was found to be spontaneous brain activity that's always occurring. So it's kind of like the background on top of which all these task-based activations are riding. And so it turns out that regions that are functionally related exhibit coherent hemodynamic fluctuations under these so-called resting state conditions. And so, for example, if I were to place a seed or an ROI and look at the activity in that left visual cortex using optical methods, we can record changes in oxyhemoglobin. Now, if we place a seed in right visual cortex, we see that the fluctuations are very temporally coherent. So that if we just take a simple dot product, Pearson-R correlation coefficient, we see that these two regions are highly correlated. And therefore, functionally related. Now, conversely, if I place a seed in motor cortex, you see that its hemodynamic fluctuations are completely different than left or right visual cortex. And so if you take the dot product there, you see that the correlations are very close to 0. So these are correlation coefficients for one particular voxel in the brain. We can then make a map of these correlations and see patterns that look like this. So this is resting state functional connectivity analysis with a seed placed in visual cortex. And so the patient is just sitting there, silent, doing nothing in the bore. And you see these very distinct patterns. Now, if they flash a checkerboard in their eyes at these fixed intervals and you look at what areas activate, you see very strong correspondence between these two patterns. And so this technology or analysis of looking at resting state functional connectivity kind of opens your patient population up to people that can't normally perform any tasks. So people in comas after stroke, babies, etc. And so you can look at a bunch of different regions. So somatomotor, other regions that aren't bilaterally distributed, like the frontoparietal attention networks, and you see very strong resemblance between those engaged during task and those found to be correlated functionally at rest. And so these resting state functional connectivity patterns provide an excellent biomarker of brain function. So you see these very distinct patterns in different networks. But this pattern disruption are often hallmarks of some type of focal disease like stroke or neurodegenerative disease like Alzheimer's. And so what we want to do, and like what we've been kind of motivated to do over the last 3 years, is put technology like this in a mouse model. Because in mice you have a whole bunch of different genetic manipulations, molecular manipulations, invasive manipulations, all kinds of things that allow you to investigate why these patterns are there and why are they disrupted. And so what I've been doing since probably 2011 is co-developing this technology called functional connectivity optical intrinsic signal imaging. And so compared to some of the systems that you guys might use, It's relatively simple. So it's a camera that's out of this field of view, a lens, and then an LED ring. And we just shine multiple colors on the exposed mouse brain, or rather, the exposed skull. So the skull's left intact. And so we illuminate with red, orange, yellow, and blue. And because oxy and deoxy absorb light at different wavelengths, different amounts, you can unmix the contribution to your optical intrinsic signal into the oxyhemoglobin, deoxyhemoglobin in total. And so our camera's running at 120 hertz. And so our spectroscopy is done at 30 hertz. So we're sampling at very high temporal resolution, these hemodynamics. And like I said before, the skull's left intact. And so we analyze these movies of the brain kind of at the pixel level. So for a given time trace or a given image sequence, we look at this pixel, perform our spectroscopy, and then it allows us to look at, say, oxyhemoglobin. So here's a power spectrum of oxyhemoglobin. And because of our high temporal resolution, we're able to fully resolve pulse and breathing. And you see this kind of 1/f fall off. So there's a lot of power in this lower frequency band. And so we filter over the functional connectivity bandwidth and then resample to a hertz. So every 5 minutes of data we collect is a little over a gigabyte. So these are giant data sets. And so we downsample mostly because we're looking at these lower frequencies, so a lot of the data are just wasted. So it makes our data sets more manageable. We perform some other things similar to fMRI, but when all is said and done, we have these movies of oxyhemoglobin or deoxyhemoglobin, our total. And so what you're looking at is the spontaneous activity in a mouse brain while it's sitting there in our imaging system doing nothing. So it's anesthetized with ketamine, a ketamine-xylazine mixture. The movie's sped up about 20 times. These are really, really slow fluctuations, some of which take 100 seconds to complete one oscillation. But this is what's happening in the mouse brain. This is what's happening in your brain. Maybe a little bit more complex, but you get the idea. And so similar to the fMRI analyses I showed you, we look at these movies and look at what's happening at different seed regions in time. So if we look at what's happening in the red, green, and blue seed, you see these 3 time traces over 5 minutes. And so you see that what's happening in the green and blue seeds are very temporally coherent. They're kind of doing the same thing and doing something completely different than the red seed, which is placed in motor cortex. So if I take this green time trace, yeah?
How many wavelengths do you use to generate that map?
We use 4 LEDs, red, orange, yellow, and blue. But we use the full spectrum in our spectroscopy inversion. So if I take this green seed and compute the Pearson-R correlation coefficient with every other time trace in the brain, we see this functional connectivity map for that green seed. And you can see that the region containing the blue and the green seed here, retrospinal cortex, is very functionally distinct and isolated from the rest of the brain, specifically that in motor cortex. So you can do this type of analysis in kind of the 8 major brain regions within our field of view, and you get these very distinct functional connectivity patterns in mice. So these maps are the average of over 10 mice, but even within 5 minutes of data, you get maps that look very comparable to this. And so kind of the classic way of validating functional connectivity from an fMRI perspective is to compare these functional connectivity maps with those during evoked responses. So if you electrically stimulate the left and right forepaw, you get these very clear activations that overlap with forepaw somatosensory cortex. And if you then let the mouse do nothing, you don't stimulate its paws, and you perform the same analysis, you see that these seed-based analyses kind of recover the functional connections of left and right somatosensory cortex. But the cool thing you can do in mice is buy mice that are genetically engineered not to have a corpus callosum. So the corpus callosum is a thick bundle of fibers, white matter tracks that connect the left and right hemisphere. So these are genetically engineered to not have that connection. So the hemispheres kind of stay very separated from one another. And so the patterns you see in normal mice, which are very bilaterally symmetric, no longer exhibit that symmetry. So they're very unilateral. So these two things kind of prove some level of validation that these signals are neuronal in origin and not just vascular. So there's a lot of cool things you can do with these movies, right? So one of the things, and I actually didn't do this, a graduate student in our lab named Brian White developed this iterative parcellation, was to look to see if that spontaneous activity, those movies, can help kind of give a sense of the underlying functional architecture of the brain. And so the algorithm starts with an initial guess as to what's happening. So you can either have seeds based on anatomy, the singular values are the principal components of those movies, or you can just simply tile the brain randomly into squares. So for either case, you'll create a time trace that's the average of all the time traces within that initial assumption. You'll correlate each seed with every cortical pixel. Then you'll assign pixels to parcels with which they correlate more strongly. Then you'll iterate through this algorithm until the movie or the algorithm converges, until this parcellation is unchanged. So it's a lot of words. If you just look at the movie, it might make more sense. So we start with an initial condition, which are the singular values. We ask for the first 20 singular values in this movie. That's the initial condition. And then we just let the algorithm go. So it converges into these very distinct patterns. Now, if we compare that converged pattern with the underlying histology, so this is a map based on the Paxinos atlas, you see very strong correspondence between what's happening sort of in the spontaneous activity of the underlying hemodynamics with what's happening with anatomy. And so other things you can do, and so I made this probably last year, and so we wanted to ask a very data-driven type of question. So if we just march this pixel or this seed anywhere we want in the brain, how similar are those maps? Can you define functional borders based on when the similarity changes? And so I'll let it play, hopefully. Okay, and so what we're doing is we're just going to march this black seed down, anterior, posterior, and calculate the functional connectivity patterns for that seed. So we now generate this giant matrix. So it's a matrix of every pixel's correlation with every other pixel. We then calculate the similarity, which is just asking the question, how similar is, say, this visual map or that motor map to all other functional connectivity maps in the brain? So regions that are red look a lot like that map. Regions that are blue look exactly opposite to that map. And regions that are green and yellow look nothing like that map. So you can kind of see these very distinct transitions in similarity. And so we then take the gradient of this map to get this pattern, and then using a simple Canny filter, finding the edges of this gradient map, and we get these borders. So then we do this for 25 mice. And we get these very distinct functional borders from the resting state functional connectivity that corroborates really strongly with what was done with this average parcellation scheme, both of which look a lot like the underlying histology. So back to kind of where the focus of the talk is, we see these very distinct functional connectivity maps in mice. And so the point of this slide isn't to say that these maps look like these maps. The point is mice have them. So in humans, these patterns are disrupted in almost any disease I can think of. And so we want to look at functional correlates of that disease and then try to use these maps to evaluate some kind of therapeutic intervention. And so the first application of this technology was done in late 2012 with Dave Holtzman's group, who's a predominant Alzheimer's researcher. So I assume some people have heard of Alzheimer's disease, but maybe don't know the specifics of it. But it's the most common form of dementia, and it's characterized by these structural and functional hallmarks in neural networks. And so one of the things that's predominantly found in AD patients are these plaques. And so if you look at the plaque patterns of this amyloid deposition, it kind of looks rather random. It kind of looks like it's just spread out all over the brain. But if you look at the pattern of this deposition, and then look at one major brain region in the human brain called the default mode network, you see that there's really strong correspondence of this deposition pattern with this default mode network. And so this default mode network is kind of this unique region that exhibits high resting state metabolism, so it's very active. It's very energy demanding from a network perspective. It exhibits high functional connectivity magnitude, so the fluctuations in these brain regions are very temporally coherent. And then it exhibits a high number of functional connections. So not only is it big, but it's also well connected. There's a lot of densely packed functional connections. So there's this really interesting relationship between amyloid beta deposition and functional connectivity. So regions that are highly connected tend to exhibit more vulnerability for plaque deposition. And then once there's plaques, that tends to deteriorate or disrupt this underlying functional connectivity. So this bidirectional relationship was something that our lab and the Holtzman group wanted to study in mice. And so we looked at male ABP PS1 mice kind of before and after they were supposed to develop plaques. And so the young PSAPP mice that exhibit no plaques show very distinct normal functional connectivity patterns that hopefully at this point you know and love. And then when we image them at a year, you see that there's complete disruption in a lot of the networks. And so this is kind of a lot of data to look at once. So one of the ways that we compress these maps to kind of characterize the disruption is to look at bilateral connectivity. So for example, look at the connectivity between left frontal cortex and right frontal cortex. So a single number. So we're going to press these entire maps into a single number, and we're going to plot it. And we see here that frontal cortex is significantly disrupted in this age group. And we're going to do that for these 6 brain regions and see that all of the regions exhibit disrupted functional connectivity. It lowers, but some are disrupted more than others. So then we do histology and kind of characterize plaque burden in these animals. And you can see that retrosplenial cortex looks like there's a lot of plaques. Cingulate looks like it has a lot of plaques, but other regions like visual don't harbor a lot of plaques. So we'll characterize them, and then we'll plot plaque load as a function of functional connectivity decline. And so regions that don't exhibit a lot of plaques also don't seem to have a lot of functional connectivity disruption, whereas regions that harbor a lot of plaques are more affected. But kind of the cool thing that we found was that if you look at the bilateral connectivity in the young mice, so you kind of see this envelope pattern in the connectivity. It seems to look a whole lot like the ultimate plaque deposition pattern in the age group. So this is almost 8 months down the road. And so what we found was that functional connectivity in the young mice was highly predictive of where the plaques were going to form and how much plaques were going to be there. So you might ask, well, okay, what happens to just normal mice if you age them from 3 months to 12 months? Do you see the same types of decline? The answer is no in most regions, except for retrosplenial cortex, which is kind of interesting because it's a memory area. And so we see a decline in retrosplenial cortex functional connectivity, but not as much as we saw in the Alzheimer's group. So kind of the main conclusion was that the functional connectivity decline in retrosplenial cortex is associated with normal aging, but then made worse by amyloid deposition. And so since 2012, we've had a number of follow-up studies with the Holtzman group, treating animals that already exhibit plaques and helping to clear some of them and also restore some function, restore some behavior. And then another gentleman, I couldn't possibly tell you what this study's about, but it does involve the circadian clock and how it's affected by plaques. And so we're going to kind of change gears here. So Alzheimer's disease is a very diffuse disease. It affects a lot of brain regions in different ways. Kind of what pays my bills and what I've been working on for the last couple of years is looking at stroke and stroke recovery. And so similar to AD, there's kind of this network level disruption from stroke. And functional connectivity between these bilateral or homotopic regions is significantly affected after stroke, as is behavior. And so we wanted to look at this relationship, the relationship between stroke damage and functional connectivity in our models. And so we trained mice to remove tiny stickers from their forepaws. We then imaged them, waited a few days for their scalps to heal, gave them a stroke, retested them over days 1 through 3, and then re-imaged them again. And then after they were all imaged and all the behavior was collected, we harvested the brains and stained them with TTC, and then separated them into 3 groups. And so the 3 groups are blue, orange, and red. So blue are all subcortical strokes, shown here. Group 2 are subcortical with some lateral cortex involved. And then group 3 are kind of these very large strokes involving cortex and subcortex that can exhibit like a lesion over almost the entire hemisphere. And so you can kind of see within our field of view of the camera when we can see the lesions. So we actually don't ever see the lesions in these group 1 mice. But we do start to see some sense of a lesion here, and then group 3 mice, again, you have this lesion over the whole hemisphere. And so if we look at the spontaneous activity in the brain, in the non-infarct or relatively healthy tissue versus the infarct tissue, you see these features. So not only are the high-frequency components of the spontaneous activity attenuated, but you also start to see this kind of phase delay. So what's happening on the healthy side is also happening a little bit in the infarct side, while attenuated and then time-shifted. And so kind of as a function of stroke severity, you start to see these greater and greater time lags exhibited by the spontaneous activity in regions associated with the lesion. And so if you stimulate the forepaws and looked at the evoked response in left and right cortex, for the most part, the response in the healthy limb is relatively unaffected. It does look like it focuses a little more, but the magnitude is roughly constant. But you see this very gradual decline in the right forepaw, the affected limb. And that decline, that reduction in the evoked response is strongly correlated with infarct volume, whereas in the left forepaw, it's not. And so, okay, let's look at the functional connectivity patterns. Let's see how much they're affected. And so these are the control group before any stroke procedure. And then even in group 1, with these only subcortical strokes, so this lesion's out of our field of view, but you start to see these very distant effects. So this is somatosensory cortex, so it's kind of right here in this slice. And you also see some disruption in visual cortex. And now, as you might expect, as the strokes get bigger, kind of the damage gets bigger. But some regions are more affected than others. So like the AD work, we're going to compress these maps into bilateral connectivity values. And you can kind of see the gradual decline that occurs in motor cortex and retrosplenial, whereas somatosensory and visual are kind of damaged right away, even with the smallest lesions. So instead of looking at single numbers, you can make maps of this bilateral connectivity. And so by definition, bilateral maps are symmetric. We're choosing to visualize them in the ipsilesional hemisphere. But you see in the control group that there's a lot of differences in bilateral connectivity. So motor cortex tends to be very highly correlated. Retrosplenial cortex tends to be very highly correlated, highly connected. And other regions like somatosensory aren't as connected. But you start to see that with bigger and bigger lesions, that sort of this brain-wide disruption in temporal coherence gets worse and worse with these larger strokes. And so we find that disruption in somatosensory cortex isn't at all correlated with infarct volume. So small lesions do as much damage as big lesions. Whereas other regions like motor and retrosplenial, you start to see this graded decline in functional connectivity with infarct volume. So if we look at the behavior, we see that each group took longer and longer to remove the adhesives. But interestingly, we didn't see any correlation with somatosensory, so it's related to touch and feel of the paws, or feel of the stickers on the paws. But motor cortex, so the ability of the animal to actually move its paw to its mouth to remove the sticker, was highly correlated with connectivity in that region. And we can't really explain why this is. So retrosplenial cortex was the most correlated with this behavioral task. And this behavioral task isn't traditionally associated with involving retrosplenial cortex. But what we speculate, and it's complete speculation, was that the ability of the mice to remember how to move the sticker off their paw might be impaired. And that might be why this region is correlated with it. Retrosplenial cortex could also be a hub that involves a lot of integration from other regions. So we don't really know why this is, but we wanted to report it because that's what we found. And so while MCAO is a reasonable clinical model of stroke, at least in these mice, the lesion size and location varies all over the place. So there were 25 mice, and we had like 12 here, 12 here, and 12 here, 8 here, and like whatever's left here. So you don't get a consistent infarct, you don't get a consistent lesion. And if you want to characterize stroke recovery, you really want a very consistent lesion because you want to look at remapping and you want to have a very controlled type of injury. And so in order to look at stroke recovery, we went with this photothrombosis model. And so the good thing about photothrombosis as a model for stroke is that you can target very specific regions of cortex. So here we're targeting forepaw somatosensory cortex only. And if you look at the group before we gave them a stroke, you see these very distinct connectivity patterns. And after stroke, you see that only forepaw connectivity is affected. So neighboring regions like barrel cortex and cingulate aren't affected at all. So you get very significant decline in the region you're targeting with very specific behavior deficits. So it's a really nice model for looking at how does this region that you just knocked out remap to other cortical areas. And so if we look at motor, forepaw, and hindpaw in the control group, we see these connectivity patterns. We're then going to play, our lesion's going to be here. And we completely wipe out forepaw. Hindpaw in this case is actually a little bit affected. But then over time, you start to see a recovery in this area. So you see forepaw cortex kind of comes back surrounding that lesion. And so, and hindpaw kind of also has this crescent shape around the lesion. And we can draw contours over connectivity values equal to 0.3. So we're just drawing contours to show you where these maps remap. And so forepaw straddles the lesion, hindpaw kind of goes around it. So that was nice to see that we have a consistent model of functional recovery. But we want to ask some questions. So we know that restoration in functional connectivity appears to be a consistent proxy for recovery. But it's been found through various studies that activity in these functionally related brain regions is thought to affect stroke recovery. So for example, contralesional activity can negatively affect plasticity. Again, there's time windows with these things, but there is an effect here from the other side, the healthy side of the brain, and how it impacts the lesion side. Results from transcranial magnetic stimulation and other methods are mixed, probably because patients don't have the same stroke across the board. You know, you get a patient population of 30 people, you have 30 different strokes. Some come in 2 days after stroke, some come in 20 years after stroke. And so there's a lot of reason, like there's a lot of, it's not surprising that TMS studies are kind of mixed in this result, and like how they affect brain plasticity and the ability of that region to recover. And so we want to ask this question in mice, can we manipulate network activity? And we chose to use a method called optogenetics. And so many of you may have heard it, but I'll just bear with me if you haven't. So it's a light-gated ion channel. So it was derived from algae. You place it in the genes of neurons. And so when you shine blue light on these neurons that express this, in this case, channel rhodopsin, they will fire. They'll fire. And so you can cause neurons to fire. You can cause changes in activity by just stimulating them with blue light. And so the excitatory optogenetic construct is called channel rhodopsin, so it depolarizes neurons. So see, if you just look at electrical recordings, the neurons are relatively silent, and then you shine blue light on them, they start firing. You turn the light off, they stop firing. And the opposite happens with a construct called halorhodopsin. So it's an inhibitory construct or a hyperpolarizing protein. And so you can do cool things with these. You can package them, the opsin, the channel rhodopsin or the halorhodopsin, into particles that the viruses carry. And so you can express them under a given promoter, so you can target very specific cell types. And so we decided to do this in a small group of mice. So 5 mice were injected with a virus carrying CAMK2, so it's targeting excitatory pyramidal neurons with this channel rhodopsin construct. These are tagged with YFP, so you can see where they go. That's this image here. So it actually spreads, it transfects a pretty large region. Depending on the number here, that'll govern kind of how widely spread that is. But we wanted to make sure we hit a region and that we could see its effects. And so you can see the expression at the individual cell level of this YFP, and therefore of this channel rhodopsin. And so for this very simple initial design, we just stimulated the mice for 10 minutes. So it was a very traditional block design, 5 milliwatts of laser power, 10 milliseconds of pulses delivered at 10 hertz, 5 seconds with no light, 10 seconds of light with this paradigm, 35 seconds off, repeat. So we get very robust hemodynamic responses from this stimulation. So you can see the blue lines here. This is when the light's on. So you shine the light, turn it off, you get this very pronounced hemodynamic response. So these are traces of oxy-deoxy in total. This is actually the average of, I think, 4 mice, but you get traces with this fidelity in single mice, single trials. And you get very distinct activation maps. So the same types of maps that you'd see by stimulating, say, forepaw cortex or somatosensory cortex, you can get by stimulating with optogenetics. I would like to show something that I took out, but I've since changed my mind. So in this same group of mice, we wanted to look at, can you modulate functional connectivity? So I blocked out this region here. So when they do the injections, they have to do a small craniotomy. They have to inject. They put a wax cap on. So that completely gets in our way of seeing what's happening in the cortex. So I've just masked out that region. That's the region where the injections were done. This blue circle is where we're going to shine light. So this is the before group. So these are the average bilateral connectivity maps that we've seen before in these 5 mice. And so when we stimulate, we completely ablate the communication between left and right, in this case forepaw cortex, to a very significant degree. So we're basically just turning off communication between those 2 regions. So that's during the laser stimulus. So we're only looking at epochs where there wasn't laser stimulation after the blocks. So now, if we turn the laser off and look for the next 20 minutes, we see that by stimulating in this region, which is left forepaw somatosensory cortex, that we see significant increases in functional connectivity in regions that have nothing to do functionally with that region. So we see increases in retrosplenial, increases in parietal, and some increases in visual. So let me get back online here. Okay. So we learned kind of a lot doing these stroke studies. So in general, the paper that I showed you of my work in stroke, Swiss Webster mice gain a lot of weight. They become very reluctant to go down under anesthesia. And then you end up overdosing them and they die. So we've gone to C57 mice for some of our studies, but talking to Ron, that might not be the best course of action. And then these repeated surgeries in the stroke recovery experiment. So we're going to image them before stroke, give them a stroke, and then image them at week 1, 4, and 8. Well, that's a lot of surgeries. There's a lot of scar tissue that builds up on the skull. And so that gets in our way of imaging with this technique. And so for any of these longitudinal studies that we're going to do again, we implanted these cranial windows that we'll see in a second. And then with the optogenetic stuff, especially with the targeted viral delivery, as awesome as it is, you have to inject lots of regions if you want brain-wide expression of this, of the ability to stimulate different regions. And so now you can just buy mice that express these constructs all over the brain. And like every week you get cooler and cooler models. And so we're actually breeding mice to express channel rhodopsin under certain promoters in certain brain regions. And then as soon as we had the cohort ready to go, you could just go buy them at Jackson. So it took us a year to make these mice, and now you can just buy them for like $50. And so if you want to stimulate multiple sites, you want to target different regions in an automated fashion. So there's some system developments here. There's some just protocol developments here. All of this stuff we kind of learned as we went. And so we're going to now just focus, and it's almost over, so don't worry. We're going to look at the cranial windows, and then for these longitudinal imaging studies, what we really want to do is develop an awake prep. So a way to image these mice while they're awake. So we want to stimulate a bunch of different regions of the cortex. Ketamine anesthesia only lasts for about an hour. Isoflurane anesthesia doesn't work at all, really. So it ablates any kind of spontaneous activity in the brain, even under the lightest of doses. And so we developed this awake prep. So the mice, so here's LEDs shining on the head, red, green, and blue. Simple camera lens setup. But the mice are head restrained, and they're running around on this ball. So this ball is levitated by pressurized air. And while we image, the mice are just running around on this ball. So it's kind of this like spherical treadmill. So we completely stole this idea from David Tank's group out of Princeton, who was doing 2-photon imaging in mice as they navigate virtual environments looking at the hippocampus. But we thought it was such a cool idea that we needed to have it too. And so this cranial window is obviously really simple. It's a piece of plastic with pre-tapped holes, and then you just metabond it to the skull. So the skull's left intact. You do the surgery as you would with the control group, or anytime before you image, and you just tuck the skin into the glue. It heals normally. The mice wake up from this procedure. They run around, they cuddle, they eat, sleep, just go about their daily business. They don't seem to be too affected with the measures that we have to measure their happiness. But they do require a bit of time to acclimate to the ball. 10 minutes a day, day one, you kind of get freaked out because their head's restrained, and you'd probably react the same way. If you were put on a styrofoam ball and then had light shining in your eyes and things like that. So you can't really blame them for being freaked out. But over the next few days, they get really comfortable. And so they take to it right away. They start running around, and they're cool. So after you acclimate them, then you can image them. And you basically get kind of these normal patterns. I'll actually show you that too. Actually, this slide kills two birds, because here's the awake patterns in the normal mice. And so they look a lot like the patterns that we saw in the ketamine anesthetized mice. What we do see that's probably not that unexpected is that we see really increased connectivity between somatosensory cortex and motor cortex. So remember, they're running around on the ball. They're feeling that ball, that styrofoam ball. It's kind of rough. They're using their limbs to run. So it's not surprising that there's increased connectivity here. But here's what happens under light anesthesia. So this is 0.5%, 1% isoflurane. You completely kill off any of the activity. But there are some networks that are a little bit more robust. And then once you high under burst suppression, you see zero activity at all. And so with this awake prep, we've looked at these transgenic mice. Sorry. And we've looked at these transgenic mice. These are mice that express channel rhodopsin under a Thi1 promoter. So they're, I think, layer 5 excitatory neurons. It's kind of all over the brain. But you chose, in this case, we chose left whisker barrel, stimulated it, and you see this very robust activation in these transgenic mice. Now, remember, they exhibit channel rhodopsin all over the brain. So we can target any brain region we want. But you see with the stimulus that you do pick up kind of a secondary representation of somatosensory cortex near motor cortex. And with tools developed by the Allen Institute, you can kind of see, what is the layout of the brain? What are the actual anatomical structural connections of the brain in this region? And you do see, in fact, a representation here in motor cortex that's corroborated with our imaging. And there does seem to be something down here near the superior colliculus that may or may not be true, but judging from the targeted viral approach that the Allen Institute is using, there does seem to be some connections with distant sites like left forepaw, or left somatosensory. And so this is the final slide, and we're trying to improve our ability to to manipulate stroke recovery. And so we want to know, like, does network activity positively or negatively affect stroke recovery? Which networks are more influential? Are there different subtypes of neurons that affect recovery in a good or bad way? And then is there an optimal time window or duration for this intervention that we want to do following stroke? So with that, I want to thank everybody that's helped me on these projects. So this is not the Adam Bauer show. This is the work from like all of these people that we've collaborated with. And so it's been an amazing experience so far, and I can't wait to keep going. So Joe's my PI. I work in Joe Culver's lab. He's a fantastic mentor. Jim Lee is my co-mentor on my K. Andrew Kraft's a phenomenal graduate student. All these technicians are wonderful. The Brucas lab has helped with all the optogenetic stuff early on. The Holtzman lab is the Holtzman lab. They don't need me. And then Mark, Rachel, and Avi are just there for general discussion and bouncing ideas. So thanks a lot again to Aman and Bruce for having me, and I'll take any questions if you have them.